Welcome friends to a new session on molecular biology. In this session, we will discuss the role of chromatin structure and regulation of transcription. Does chromatin structure play an important role in regulation of transcription? Before discussing this, let us study the structure of chromatin. And before going into this, it is important to know various techniques which led to the chromatin discovery. It will be seen that the progress in the study of chromatin structure in early years did not make much headway due to limitation of techniques. Here we will discuss the structural details and the organization of the nucleosome subunits. In the year 1960, Maurice Hugh Frederick Wilkins and Vittorio Luzzati, while studying X-ray diffraction patterns of chromatin, observed repeating units in chromatin. Similar repeats were inferred from X-ray diffraction of a mixture of DNA and histones also. In 1974, Ada Levi-Ollins and Donald Edward Ollins, who were research professors in University of New England, reported the results of electron microscopy of chromatin obtained from interphase nuclei lysed in water. Under electron microscope, chromatin fiber appeared as arrays of spherical particles about 10 nanometer in diameter connected by filaments which were about 2 nanometer in diameter. These particles were called new bodies. The structure of DNA suggests that DNA alone cannot form repeats at intervals of 10 nanometer, which meant that the association of histones must have conferred the repeating organization to chromatin. Association of histones is also suggested firstly from the knowledge that histones are positively charged and will associate with negatively charged groups in DNA, and secondly from the knowledge that histones perform their function in chromatin by binding to DNA. Therefore, it was suggested that repeating units in chromatin result due to association of DNA with histones, and that these repeats occur at intervals of 10 nanometer. Let us examine the basic structure of a nucleosome subunit of chromatin. The diameter of nucleosome is 11 nanometer and its height is 6 nanometer. The length of DNA around it is 70 nanometer, which is equivalent to 200 base pair. Approximately 67 nanometer DNA is approximately to 200 base pair, because 34 Armstrong units is equal to 3.4 nanometer, which is equivalent to 10 base pairs. The length of DNA varies not only in different organisms, but also in different tissues or even in different regions within the same cell of an organism. The length of DNA, however, suggested that DNA should be coiled around the nucleosome core particle. Enzyme DNA 1 also led to cleavage of DNA, suggesting that DNA lies on the surface of nucleosome. It was therefore suggested that histones do not cover DNA, but DNA is wrapped around histone core particle. In other words, it was suggested that it will be more appropriate to describe chromatin as string on beads rather than as beads on string. The properties of nucleosome model are presented in this table. The different constituents consist of 1 DNA, the size of which is 200 base pairs. The mass is 1,30,000 Daltons. The core particle consists of size 140 base pairs and mass of 90,000 Daltons. The second constituent consists of histones, which includes H2A, H2B, H3, H4, is octamer consisting of 2 molecules each, and the mass is 1,08,000 Daltons. The core particle consists of octamer consisting of 2 molecules each, and the mass is 1,09,000 Daltons. The third constituent is H1 histones. The size is 1 molecule and the mass is 24,000 Daltons. It is absent in the core particle. Spiral arrangement of histones. When nuclease treatment was prolonged beyond the cleavage between nucleosomes, the DNA was removed from both the free ends, yielding particles containing DNA of only 146 or sometimes 140 nucleotide pairs instead of 200 nucleotide pairs. This reduced form of nucleosome is called core particle. Adjacent core particles, each with core DNA, are joined with each other through DNA segment called linker DNA, which is removed during prolonged digestion. The core DNA has an invariant length of 146 base pairs, but the linker DNA can vary from 8 to 114 base pairs, thus accounting for variation in DNA length of nucleosomes, that is between 154 to 260 base pairs. The closeness of 2 superhelical turns of DNA on the histone octamer also has a functional consequence. This allows the binding of a protein molecule simultaneously to 2 different sites of DNA 80 base pair apart. In eukaryotes, it will be seen that a promoter site, that is CAAT box, may lie 80 base pair upstream from start point. In such a situation, superhelical turns in the nucleosome may allow the association of RNA polymerase enzyme to the promoter and initiation site simultaneously due to closeness of the 2 turns on the nucleosome octamer. Some histones function as spools for the thread-like DNA to wrap around. Histone proteins are of 2 types, core histones and linker histones. Core histones are H2A, H2B, H3 and H4. 2 of these core histone proteins assemble to form 1 octameric nucleosome core particle, and 147 base pairs of DNA wrap around this core particle. H2A is important for packing DNA into chromatin. Since H2A packages DNA into chromatin, the packing process will affect gene expression and also found to regulate gene expression. H2A has been correlated with the DNA modification. H2A also plays a major role in the determination of overall structure of chromatin. H2B is also involved with the structure of nucleosomes of the beads on a string structure. H3 is involved in the structure of chromatin in eukaryotic cells and featuring a main domain and a long and terminal tail. It is an important protein in the emerging field of epigenetics, where its sequence variants and variable modification states are thought to play a role in the dynamic and long term regulation of genes. Histone H4 is involved in the structure of chromatin in eukaryotic cells. H4 is a structural component of the nucleosome and is subject to covalent modification including acetylation and methylation. Linker histones include H1 and H5. Histone H1 is one of the 5 main histone protein families which are components of chromatin in eukaryotic cells. Though highly conserved, it is nevertheless the most variable histone in sequence across species. H5 histones are individual proteins involved in the packing of specific region of DNA. Effect of chromatin structure on transcription. The discovery of nucleosomes impedes transcription in vitro, and that deletion of histones or their basic tails elicits specific effects on gene expression in vivo, provided a glimpse of chromatin's importance. These findings provide the pivotal roles of non-histone proteins in regulating chromatin structure. New approaches for manipulating chromatin both in vitro and in vivo. The chromatin structure has profound effects on DNA related metabolic processes such as transcription, DNA repair, etc. In prokaryotes and eukaryotes, the mechanisms underlying transcription are similar except the complexity of the transcription machinery in eukaryotes. In eukaryotes, the transcription starts by binding of the typical enzyme RNA polymerase II of the core promoter region, including the transcription start site and Tata box. This leads to the binding of general transcription factors. We discussed 7 transcription factors: TF2D, TF2A, TF2B, TF2E, TF2F, TF2J and TF2H, which are essential for transcription in eukaryotes. Several hundred different transcription factors have been found to date, 2/3 of these falling into 4 recognizable groups, that is helix turn helix, helix loop helix, zinc finger and leucine zipper. The remaining 1/3 may be unique to their types. Each transcription factor has separate domains for 1. Recognition and binding a specific DNA sequence, and 2. Activation of obligatory proteins. RNA polymerase II is positioned at the core promoter region by a combination of transcription factors TF2D, TF2A and TF2B to form the pre-initiation complex. TF2H then melts 11 to 15 base pair of DNA in order to position as template in the RNA polymerase II open complex to initiate transcription. The carboxy terminal domain of RNA polymerase II is phosphorylated by the TF2H subunit during the first 30 base pair of transcription and loses its contact with the GTFs before it proceeds on to the transcription elongation. Meanwhile, the phosphorylated CTD begins to recruit the factors that are important for productive elongation and mRNA processing. The packing of nucleosome affects all the process in transcription, from binding of the core promoter region, including the transcription start site and Tata box, to the formation of pre-initiation complex. Let us discuss how chromatin structure is regulated and affects transcription. Modification of histones. Histones undergoes modifications which include methylation, methylation of arginine residues, phosphorylation of serines and threonines and acetylation. The table further shows position of modifications, the enzymes involved in this in various organisms such as Saccharomyces cerevisiae, Saccharomyces pompeii, Drosophila and mammals. The recognition modules and functions of this in transcription are shown in the columns towards right side of the table. The modifications that are associated with active transcription, such as acetylation of H3 and H4 or di or trimethylation of H3K4, are commonly referred to as euchromatin modifications, and modifications in inactive genes or regions such as H3K9Me and H3K27Me are termed as heterochromatin modifications. Heterochromatin are tightly packed chromatin that is not actively transcribed. Euchromatin are loosely packed chromatin that is easily accessible by transcription enzymes and actively transcribed. Most modifications are distributed in distinct localized patterns within the upstream region, the core promoter, the 5' and 3' end of the open reading frame. The location of a modification is tightly regulated and is crucial for its effect on transcription. For example, SET2 mediated methylation of histone H3K36 normally occurs within the ORF of actively transcribed genes. However, if SET2 is mistargeted to the promoter region through artificial recruitment, it represses transcription. Acetylation of histones occurs at multiple lysine residues by a variety of histone acetyl transferase complexes, in short called as HATs. Distinct patterns of lysine acetylation on histones have been proposed to specify distinct downstream functions, such as the regulation of co-expressed genes. In contrast to acetylation, histone methylation, phosphorylation, ubiquitination, etc. are often catalyzed by a specific enzyme at a specific site and result in unique functions. Histone modifications have a number of different functions. Histone modifications result in a change in the net charge of nucleosomes, which could loosen inter or intranucleosomal DNA histone interactions. It is well accepted that protein modifications directly influence higher order chromatin structure. Regulation of transcription. Transcriptional regulation is a vital process by which cells regulates the transcription and enhance gene activity. Transcription regulation is significantly more complicated in eukaryotes than prokaryotes by the presence of introns and proteins involved or packing of DNA into histones. A single gene can regulate transcription and gene activity in many ways. Transcription regulation in prokaryotes. The early understanding of transcription regulation came from prokaryotic organisms. The regulation of transcription in prokaryotes is governed by three main elements: promoters, enhancers, and the positive control elements that bind to DNA and initiate transcription. Promoters are regions of DNA that bind to RNA polymerase and initiate transcription. The promoter region is recognized by the enzyme RNA polymerase and sigma factor and inhibits the initiation of transcription. In prokaryotes, transcription regulation is dependent on the strength of the promoter region and the presence of repressors and activators. The sequence based affinity of a promoter for RNA polymerase varies in the absence of regulatory elements, which results RNA transcript in different amounts. The variable affinity of enzyme RNA polymerase for different promoter sequences is related to the -10 and -35 regions of consensus sequence upstream of the start site of the initiation of transcription. In positive regulation, a transcriptional activator protein binds to the operator region to promote transcription. There is no transcription in the absence of the activator protein. In negative regulation, a repressor binds the operator sequence to prevent transcription. Only in the absence of repressor protein can the gene be transcribed. The default state of a prokaryotic transcript is to be in the on configuration in the absence of other regulatory elements, resulting in the production of some amount of primary transcript. This means that the regulation of transcription in the form of positive control elements and protein repressors can either decrease or increase transcription. Repressors often physically occupy the promoter region, occluding RNA polymerase from binding. Alternatively, a polymerase and repressor may bind to the DNA at the same time, and the repressor prevents the opening of DNA double helix for the initiation of transcription. This strategy of control is different from transcription in eukaryotes, whose basal state is to be off and the cofactors required for initiation of transcription are highly gene dependent. Transcription regulation in eukaryotes. The complexity of transcription regulation in eukaryotes carries with the complexity of three RNA polymerases known as polymerase I, polymerase II, and polymerase III. Each polymerase has specific targets and has independent mechanism in transcription regulation. The polymerase activity in transcription regulation can be controlled by three mechanisms. 1. control over RNA polymerase access to the gene. This includes the functions of histone remodeling enzymes, enhancers and repressors and transcription factors. 2. Productive elongation of the RNA transcript. Once RNA polymerase enzyme is bound to a promoter, it requires transcription factors to the promoter complex and begin transcription. Third, termination by RNA polymerase enzyme. The transcription factors controls how and when termination occurs. The difference in the generation of RNA transcripts in prokaryotes and eukaryotes is the recruitment of transcription factors. This difference is mainly due to the compaction of genome in the eukaryotes by winding of DNA double helix around histones to form higher structures. This makes the gene promoter inaccessible without the assistance of transcription factors, and chromatin structure is a common site of regulation of transcription. Similar to the sigma factors in prokaryotes, the GTFs in eukaryotes are required for all transcription events. They are responsible for stabilizing, binding and opening the DNA double helix to allow the RNA polymerase enzyme to access the DNA template. Once RNA polymerase is successfully bound to the template DNA, it often requires the assistance of other proteins in order to leave the stable promoter complex and begin transcription elongation. This process is called promoter escape and it slows the transcription process. Similarly, nucleic acid and protein factors can associate with the elongation complex and modulate the rate of transcription at which the RNA polymerase moves along the template DNA. Regulation of transcription at the level of chromatin state. In eukaryotes, genomic DNA is highly compacted by winding the DNA around protein octamers called histones. Significant portions of genomes are silenced through histone modifications and are inaccessible to the transcription factors or RNA polymerases. The highest level of regulation of transcription occurs through the rearrangement of histones, because these processes have the ability to furnish entire regions of a chromosome. Histone rearrangement is facilitated by post-translational modifications by enzymes such as the histone methyl transferases, in short called as HMTs, histone acetyl transferases, in short called as HATs, and histone deacetylases, in short called as HDACs, etc., which add or remove acetyl groups, methyl groups, ubiquitin and phosphates. Histone modifications serve to recruit other proteins which can either increase the spacing between histones or allow the association of transcription factors or RNA polymerase on DNA, or to increase the compaction of the chromatin or promoter elements. Regulation through transcription factors and enhancers. Transcription factors are proteins that bind to specific DNA sequence in order to regulate the gene expression. Transcription factors function through a wide variety of mechanisms. They can be divided into activators and repressors. Activators can interact directly or indirectly with the core complex of transcription through enhancer binding. Repressors recruit co-repressor complexes that lead to transcriptional repression by condensation of chromatin in enhancer regions. Repressors may also function in allosteric competition against activator to repress gene expression. Overlapping DNA binding motifs for both repressors and activators induce a physical competition to occupy the site of binding. In the presence of the repressor, transcription would be effectively blocked if the repressor has a high affinity for its motif than the activator. Enhancers or cis regulatory elements, otherwise cis regulatory modules, are non-coding sequences of DNA containing multiple repressor and activator binding sites ranging from 200 base pair to 1 kilo base pair in length and can be 5 prime upstream to the promoter region or within the intron of the regulated genes. Transcriptional regulation are mediated by DNA looping, which brings together transcription factors, enhancers, promoters and RNA polymerases to regulate gene expression. Through DNA looping, active enhancers binds the promoter region dependently of the core DNA binding motif promoter specificity. It provides the basis for the functional interaction between transcription factors and transcriptional core complex to trigger RNA polymerase II to leave from the promoter region. The configuration of eukaryotic genome is essential for promoter enhancer proximity. The enzyme RNA polymerase II is responsible for the production of mRNA, and much of the regulatory checkpoints in the transcription process occur in the assembly and escape of the pre-initiation complex, particularly for RNA polymerase II. A combination of transcription factors will recruit 2F2D or 2F2A to the core promoter, followed by the binding of TF2B, creating a stable complex onto which the rest of the transcription factors can assemble. This complex is relatively stable and can undergo transcription initiation in multiple. After the binding of TF2B, TF2D and RNA polymerase II, the rest of the GTFs can assemble, promote the transcription and serve as sites of recruitment for processing mRNA. Now, let us summarize the following discussions on effect of chromatin structure and regulation of transcription. Transcriptional regulation is a vital process by which cells regulates the transcription and enhance gene activity. The regulation of transcription in prokaryotes is governed by 3 main elements: promoters, enhancers and the positive control elements that bind to DNA and initiate transcription. In eukaryotes of transcription regulation carries with the complexity of 3 RNA polymerases known as polymerase 1, 2 and 3. The polymerase activity in transcription regulation can be controlled by control over RNA polymerase access to the gene, productive elongation of the RNA transcript and termination by RNA polymerase enzyme. In eukaryotes, the highest level of regulation of transcription occurs through the rearrangement of histones and is facilitated by post-translational modifications by enzymes such as the histone methyl transferases, histone acetyl transferases and histone deacetylases, which add or remove acetyl groups, methyl groups, ubiquitin and phosphates. Regulation of transcription occurs through transcription factors and enhancers. Transcriptional regulation are mediated by DNA looping, which brings together transcription factors, enhancers, promoters and RNA polymerases to regulate gene expression. Now, I will give you certain assignments based on the discussions what we had. 1. Write short notes on the basic structure of nucleosome. 2. Explain the properties of nucleosome model. 3. Discuss the effect of chromatin structure of transcription. 4. Give a brief account on regulation of transcription in prokaryotes and eukaryotes. I will give you also certain references for your further reading. 1. A book with the title Biochemistry 5th edition, authored by Berg, Timosco and Stryer in the year 2008 and published by Freeman and Company, New York, USA. A second book is with the title Cell Biology 7th edition, authored by Karp in 2014, published by John Wiley and Sons, Singapore. A third book is with the title Molecular Biology of the Cell 5th edition, authored by Alberts, Johnson, Lewis, Roth, Roberts and Walter in the year 2007, and it is published by Garland Science, New York and London. A fourth book is with the title Gene 9, authored by Levin in the year 2007 and published by Johns and Bartlett Publishers, Sudbury, Massachusetts. A fifth book is with the title Biochemistry, authored by Davidson and Sitman in the year 1997 and published by Waverly, New Delhi. Sixth book is with the title Principles of Genetics, authored by Gardner and Nustad in the year 1984, published by John Wiley, New York. A seventh book is with the title Principles of Genetics, authored by Sinnett, Dunn and Dobzhansky in the year 1958, published by McGraw-Hill, New York. I will also give you certain websites for your further reference. The first one is www.bioencyclopedia.org. Second one is www.oxfordgenetics.com. We have come to the end of this session. Thank you for watching this. We will meet in another session with a new topic. Until then, bye.